Maybe it's this. This is working. So mine is working. Maybe I can share.
Hello. Good morning, everybody. Welcome to the keynote session for today. Our distinguished speaker is Markus Axer. He's a professor in the Institute of Neuroscience and Medicine in Jülich. So I looked at the map yesterday, and Jülich is almost equidistant from Aachen and Cologne, just in case you're familiar with those two places. His work is on Connectome, image processing, high-dimensional computing. We're very pleased to have Markus here.
Oh, the timer, yes. Maybe. Thank you very much for this kind introduction. Thank you very much for inviting me here. I'm really honored to talk in front of you about the connectome. That means about structural connectivity of the human brain. And I have to warn you just at the beginning, I'm an experimentalist, so I'm a physicist, and all you hear today is from my view as an experimentalist. Because I think the connectome is still at a stage where we have to understand our devices, our tools, and set up also the infrastructure to really address the entire human connectome. What you see here is a real image of our work. I will discuss today also how to get to such views. It's taken from a cerebellum. It's based on polarization microscopy, and this is something you will get to know about in the next 2 hours, 50 minutes. So let's start with some general facts. And it's important to realize that the brain, of course, it is a massive object with at the multi-scale problem. We are talking about 100 billions of neurons, and they are interconnected somehow. Each neuron can have up to 10,000 individual connections, synaptic connections to their neighbors. And we are talking about fiber lengths of between micrometer to centimeter scales, fiber calibers between 0.1 to 20 microns. And in total, we have to deal with 5 million kilometers of fibers. So that's already the challenge, right? How to deal with this universe in our brain. So in principle, we have to start and look into different scales, starting from synapses, going to at a few nanometer scale, going to individual neurons, talking to each other, setting up the next range of the micrometer to millimeter scale. These neurons, they build cell architectures in a very specific way. Mostly we don't know how and why. They build anatomical structures. These structures, they have a specific function or many functions usually, and they are integrated into the entire brain of 1.5 liters of volume, right? And we are interacting as humans. So whatever we do describing the human brain, whether it is the connectome or something else, we have to take care of all these different scales somehow. And up to date, there's no single imaging technique available that might give us a good answer to our questions, right? So this means we have to discuss here today some challenges concerning the imaging techniques. I will give you a brief update of what is available today. Of course, data handling, analysis, and integration is a very important aspect, especially whenever you start to think about how to share data, how to use this data for some calculations. And finally, and I'm coming from a brain mapping institute, the vehicle we have to use, I think, to share the data are atlases. Because brain atlases provide labeled and curated data. They are clean, they are well controlled, and they are usually, whenever we share our data, coming from multimodal information. So that's the setup of today. And let me start with the imaging technique. So don't worry, I'm not going through the entire history of connectivity research, but I really like to start with some of our pioneers of modern brain research or brain mapping. And these were Corbinian Brodmann and Cecile and Oskar Vogt. The three of them, they worked around 1900, and they were the first who understood that the brain is not a homogeneous kind of something. It is built of different regions made up of different characteristics. And we will meet them occasionally in my talk. So one of the things they already realized was it makes sense to look into brain sections. So they generate thin sections, 20 micron thin sections, stain for the myelin, because the myelin is what many axons do have as an insulation layer around. And with this, they, let's say, initiated something that is called the myeloarchitecture. It describes the organization of fibers, myelinated fibers in the cerebral cortex. And they found out already at 1919 that cortical fiber architecture is region specific, right? We see basically two main orientations of fibers. As you can see here, you have the radial ones and then the perpendicular stripes. And they look different depending on where you are in the brain. Concerning the white matter, they were not really able to resolve all the individual fibers because the white matter is really dense. This gives trouble to any kind of imaging technique, even today, right? So it is really dense. So resolution of individual fibers is really hard, and it was already hard at that time. However, they managed to look individually into single sections and then provide us with drawings. And these drawings are still comparable to what we see these days with much more modern techniques and image processing techniques. And so this means it is already a good reference, but it's not the complete view because we are only looking into individual sections of an individual human being. So, and we see only drawings. These are, of course, 2D drawings. However, they also started to define some names of specific tracts. So we are now talking not only about individual neurons, but about bunches of neurons building pathways. And you might know these. We call them these days association fibers, projection fibers, commutator fibers, depending on which region is connected to what region. This is a brain map from the Vogts. So it's an original one, right? So this is how the people tend to present their conclusions on maps. This was part of their atlas, right? And it comes here like a kid's version, but this is really the original indication of different regions of the brain where the fiber architecture seemed to be similar. And this is still the idea of any kind of atlas. We try to find similarities and then cluster these similar regions to a specific kind of brain region, right? Just to make the picture a bit more complete, these days, and also the folks' days basis for an atlas was the distribution of neurons, not their connection. And what you see here, right, is that in the cerebral cortex, you can also see changes in the lamina structure of the brain here, right? So there's a change in distribution of cells, of the size of the cells, the thickness of the different layers you can see here, right? And this is what makes up a human brain. And this is also important to understand whenever we provide data about the connectome or something else from the human brain, we have to be very precise where we are and what we are, where this piece of tissue has been taken from. Because there's a huge difference in neighboring regions. So, of course, I'm pretty sure you all know the seminal paper where all this connectome stuff really started about 20 years ago, given by Olaf Spons et al. And here are only 2 sentences out of this publication. They show the basic idea of the connectome. So it is the centerpiece of brain connectivity, a comprehensive description of how neurons and brain regions are interconnected. It is the indispensable foundation for understanding how brain dynamics and function emerge from their underlying structural substrate. So this is it. Just two sentences, but we haven't solved this task yet, 20 years later. Okay, because we have to take care of many other things. If you want to understand function, you also have to understand what scales are important. Are there any hierarchies, some specific organizational property inherent to the connectivity? What about redundancy? How does the brain works if some connections fail? Resilience. How does the brain works on new inputs, on diseases in terms of plasticity? All these things change connectivity, change the connectome, and makes life even harder for us to derive the connectome from basic principles in some way. So this paper from 2005 pushed tremendously the development of diffusion MRI. This is what most of you, of course, know, right? So with diffusion MRI, we probe the movement of water molecules, and fiber structures represent obstacles in the free movement of the water molecules, and indirectly you can derive the distribution of fibers in a measurement with an MRI system. And you can do this in vivo. And this is today the only in vivo possibility to derive for the entire brain the distribution of fiber structures. To be honest, it's not completely the fiber structures, it's more the pathways themselves. So the big bundles built out of many, many neurons that are connecting different brain regions. In addition to the measurement technique, the people started to develop also the analysis tools, and also the main development, the main interpretation tool and reconstruction tool for diffusion MRI data is called tractography. So there are these days many possibilities to apply tractography to your orientational data. I'm not going into detail here because I'm pretty sure we will have this week additional talks from experts which are much closer to the tractography than I am. But let me give you some really easy ideas why tractography is currently failing in many times, unfortunately. And it is due to this fact. So given we have a brain, this is built of fibers, but we don't know their orientation, right? What we know is after the measurement, we get some information on the orientation. So this is the input. And it's a black box, basically. It just tells us it's a distribution of orientations or orientation distribution. So how to model and how to predict what's the connectivity based on these orientations? This is nearly not possible. But it is going, it's getting better and better, and the people are getting smarter and smarter doing different kind of tractography, but they are still not giving always the truth, and they cannot, because the data basis is not good enough to have a clear, realistic reconstruction of the complete connectome data. So I'm pretty sure you know it better than I do. The Human Connectome project was the project kind of blueprint for all connectome projects worldwide, how to address this kind of questions. They developed new techniques, new devices, also new tractography results, and also the sharing of the data. So that was the starting point, really the starting point for modern connectome research. However, over the years, everyone realizes that we need, on top of these kind of orientation information, real anatomical input. And this pushed further many different imaging approaches over the recent years. And this is the latest one, right? I'm also sure you've seen that being published a few months ago by the Lichtman group, and they did electron microscopy on 1 cubic millimeter of human temporal lobe. The donator was diseased, so it was a piece of tissue taken from an epileptic patient, and they applied not only electron microscopy on this, they applied a bunch of AI-based analytics for segmenting out individual neurons and also all other compartments you can see in one of those big blocks here, right? And this is the complexity you see for an individual neuron. And the question is, now thinking about, we have 100 billions of them in the brain. Do we need this kind of resolution to understand the connectome and the effect for functionality? We don't really know. I would expect, yes, and I would like to see this kind of data for the entire brain, but this is currently out of reach, right? So this 1 millimeter cube of data had already a petabyte of data to describe it. So go to 1.5 liters, you're completely lost. No way. So there are other techniques on the market, and this is an overview of what in my mind is currently driving the postmortem aspect. So trying to find really anatomical information to inform much better the diffusion MRI data for interpretation, right? So we found out that it is possible to apply diffusion MRI also to postmortem brains. So everything is dead, nothing is moving, but still it works. We have set up, so saying we means the community, right? So the connectome community is currently also using a couple of different scattering approaches. which means they're using X-ray, neutron, or visual light to be scattered in the tissue and then learn from these scattering behavior how the orientations of the fibers are. Then we do have polarization dependent analysis. So we have two main parts, polarization microscopy. This is what I'm going to tell you today in more depth, because this is my work. But there's also in Boston a very good group on optical coherence microscopy providing similar kind of data. While polarization microscopy is based on sectioning the brain and then rebuilding these sections to a volume, which is always a limitation in any kind of technique. The optical coherence microscopy can be applied before the sectioning. So you don't have the problem with the registration. On the other hand, the polarization microscopy provides a bit higher resolution in plane. So so these guys here on top, they are able to provide information about the orientation of fibers. More or less directly. More or less means we use specific physical models to describe the orientations, right? So this is physically based. That's cool for a physicist like me. But here, I've shown you already the electron microscopy, the latest results. Fluorescence microscopy, of course, is being used everywhere. So it's of course being able to be used for the human brain. And this is, I would say, the most close to the brain approach you can have. It's dissection. So you can really dig yourself through the tissue and try to follow fiber pathways. And this gives you direct information on how different brain regions are connected. But you can only dig once per brain, right? So you have to decide which kind of path you have to follow. And here, in this case, right, you receive, for these kind of techniques, you receive scalar data, and you have to interpret them first in terms of orientations if you want to describe the connectivity based on tractography. This means for all of these guys, you have to find a way how to segment the data and then how to apply structure tensor analysis, for example, to describe the orientations or gradients in your measurement and interpret them as fibers. So this gives you additional stress. But it's also all possible, and worldwide the people are doing one of those, mostly compared with MRI. So you find, if you find at all a direct comparison of measurements in the same brain, you have usually always MRI as basis, and then you can compare with one of the others. That's cool, but that's not all we should do. So I told you I'm a fan of polarization microscopy, and I'm going to tell you now why. It goes back to Brodmann. We already met this guy. He's one of the mapping pioneers. You might know the Brodmann map based on the distribution of cells. And he wrote in, when was it? 1903, the following sentence here: formalin fixation does not impair the birefringence of myelinated axons. So that's a physical effect. Therefore, we can study nerve fibers hardened and conserved in formalin with polarization microscopy and observe possible pathological alterations. And so we try to rebuild this kind of idea, use polarization microscopy and see what's happening. What do we need? We need to section the brain. So it looks like this. We freeze the brains down to -80 degrees. We put some glue around, and these blue guys here are fiducials for later 3D reconstruction, right? We always take a photograph of the top of the surface on top. And this photograph here, you see after alignment of the serial photos during this sectioning step. We are sectioning at 50 microns, so hair thin thickness, which means we end up with about 3,000 sections per human brain. Okay, and we can use this kind of 3D reconstruction already as a reference. So that was the last situation of the brain without cutting, without losing information, without destroying the tissue, whatever can happen during the preparation technique. Then we put it into what we call a polarimeter. So we have built in Jülich something that allows us to measure birefringence. And this is based on simple linear polarizers and a circular polarizer, and the linear polarizer is rotating. We have a green wavelength source, light source, and we have a camera on top. And this is what we see in a close-up here. So that's a sagittal section through an entire human brain. So that means that's 15 centimeters, right? And you can also see a little bit here, there are some kind of tiling effect to see, right? We are addressing the brain at 1.3 micron resolution in plane. So that means the field of view of our microscope is only 3 millimeters in all directions. So that means we have to measure tile by tile and then rebuild the entire panorama view of the section later on. And this is what you can see here, indicated here in these tiny tiles. Every tile has about 5 million pixels. So that's a huge data set, right? We are acquiring images under different rotation angles of the polarizing filters, which means we end up with about 200,000 individual images, about 1 terabyte of raw data per section. So here we really have to take care of how to deal with the big data we are acquiring. So this times 3,000 sections means 1 human brain makes up something like 3 to 4 petabyte of raw data. So that's really a big experiment, and it's comparable to everything we see from particle physics or astronomy experiments. So what do we do in the next step? You see here, while rotating the filters, the intensity changes, and depending on where you look into the brain regions, you see different kind of intensity changes. They are always sinusoidal, but they have different phases and they have different amplitudes. And based on these information, and also they have different means, we can derive an idea on the 3D orientation of the fibers just from doing these kind of 2D imaging. So we have built a couple of different types of polarimeters at different resolutions, from 60 micron down to 1.3 micron. And we have to deal with a lot of data. That means we had to set up in the last years a whole workflow also for high performance computing. Fortunately, Jülich has one of the biggest European supercomputer in-house, so we can use it. And we had to set up a very complex way of dealing with how to bring the data from the lab with automated QC to the supercomputing center and doing analysis of the data we have acquired. And then also providing the data back to have a view on them, right? So this is really data-driven supercomputing. We started about 10 years ago, and it took 10 years to get an idea how to really do it. So what can we do with the data then? So there's a physical model we can use to fit the sinusoidal changes of the intensities, and taking the mean values here, the green ones gives us this information of the brain section. The orange one here is the direction, we call direction. It's one of the parameters in our model, and the other one is called retardation. And you see We see 3 different types of information in one from one measurement. And what does it mean in anatomical terms? Well, we can reconstruct based on these 3 modalities. We can reconstruct, for example, the 3D orientation of the fibers based on the fitting model. And then we provide to every distinct orientation specific color, encoded as a hue and a saturation value. Right? Hue always means, so if you have clear colors, it's mostly in plane orientation, and whenever the color is getting darker, you have out of plane orientations. And with this technique, we can really not only reconstruct white matter orientations, we cannot resolve individual fibers. I already pointed this out. At this level of resolution, with this density, with this section thickness, but we can see the orientations, we can measure them, and we can also look into the cortex and see cortical fibers. So this is also new. So typically we are thinking, or the community is always thinking about myeloarchitecture. This is the architecture of the fibers in the cortex, and white matter. And they are very often not handled in the same way because of different reasons, right? But there are different locations of the same kind of connectivity, right? So we applied this technology to different species as well. So it opens up the possibility for us also to compare across species like mouse, monkey, pigeons. So birds are cool. I like the birds because the brains are smaller, so you can much easier deal with them, but you are already also in a terabyte, at the terabyte scale to provide this information. Yeah, and we had some good success on looking into these different species, comparing them, and seeing that that the rat brains and also the monkey brains, they share similar architecture as the pigeons, which was a surprise to everyone. And eventually it was one of the breakthroughs of the year 2020 in science. And it started a lot of research on the bird brains. So once I've only shown you individual sections, right? So this is already hard to attack, but doing 3D reconstruction is more challenging. It's the main problem of section-wise analysis, of course. So you have to rebuild the brains. And here is our very first proof of concept. So it's a rat brain, and it has been sectioned along the perpendicular orientation as shown here. So the movie stopped, but don't worry. And you could see that there's a coherency across the section. So that means it was the first indication that this kind of technique can be used for 3D imaging of orientations. We did it for a larger brain, for a vervet monkey brain, not for an entire one. Although we have measured the entire one, we just have 3D reconstructed pieces, bits and pieces up to now. You can see here a visualization of only of the amplitude of our measurements, and they are just aligned, and you see already scalar information of larger tracts if they are individually visible, and we can on top project now also our orientation measurements. So that's really helpful. Based on this data, we started to think about how can we describe the fiber architecture, because the fiber architecture analysis is usually taken or interpreted by means of tractography. But of course, nowadays with AI, you can do maybe much more. And we tried some kind of applications here. And eventually we found that contrastive learning helped us to extract features in the 3D space of our reconstructions to describe different characteristics of the tissue. So saying we see clusters of U fibers in a specific kind of feature component. We see steep fibers and can distinguish them from crossings and so on. So that might be a next good way to describe somehow the fiber architecture. We are not finished with this, but these are the first results on our data. The other point is, of course, we are extracting orientations, right? And so you can generate some kind of orientation statistics to be more comparable to the MRI, which means we can collect the orientations, the orientations here across section depth. We can generate from this orientation tensors, and we can also describe, so to say, the distribution of measured single orientations in a voxel that has a similar size as the diffusion MRI provides. So that we can easily rescale our high resolution data to align this data with MRI techniques without really losing the information, the high resolution information, right? And here you can see that's an example starting with high resolution information and then ending up with an already highly resolved MRI data set, right? So this is the information you have to work with if you go for diffusion MRI alone. So this shows that we have to go to the next steps. Finally, we did some first tractography approaches using different types of implementations of tractography, downscaling. It's more like a proof of concept, and we think we are able to do, let's say, tractography on the orientation data at high resolution, and we are currently preparing for using our next generation of computer. So it will be the very first European exascale computer we can make use of. It's called Jupiter. It will be usable, or pieces will be usable at the beginning of next year already. So we are trying to set up a specific tractography approach that is highly scalable and really requires exascale computing to really realize or maintain the high resolution we can provide, not having to downscale all the data. We are not sure whether this will work, but in the end, we plan to have the resources, the implementations openly available, and also for the community to use the exascale computer as well as a working kind of software on that. But that's future. So we have set up over the last 18 years, I'm in Jülich, a whole workflow to get from a postmortem brain to some fiber descriptions. But it's not enough. You need to know, you need to integrate your data. It's only one view, one resolution of data you're looking at. So you need to integrate other modalities, not seeing them as individual different patches. This is applied to this brain and the other one to the other brain. I think the breakthrough will be in the end bringing together as many different scales and imaging techniques applied to the same tissue. And this is what we are trying to go for. And of course, starting with an MRI. So this is a postmortem brain here. We are looking into a container, a specific container for diffusion MRI of one hemisphere. We did high field, high gradient MRI with our colleagues from France, from Neurospin, and also applied polarized light imaging to this and integrated the data directly. We see common features, so we can align these common features and try to understand how this works. We did this already for the hippocampus. So we have reconstructed a hippocampus based on nearly 600 sections, right? And you see here, sometimes you see incoherencies. This is due to the fact that the registration of the serial sections is not perfect still, but it can be optimized. And this data set will be published very soon, and also published in a way that everyone can download it and have a look and play with it. So we also invested time in scattering approaches. So that means correlation microscopy between polarized light imaging and what we call scattered light imaging. So the idea is quite easy. You have a light source, you change the light source during the measurement, and you are only measuring in the middle of the illumination field, right? And what you then see is an effect of scattering of light. And from the scattering, you can derive some kind of profiles, and you can derive information on top of the polarized light information, which tells you the different orientations that might be included in one pixel. So that means you get a better idea on what's the distribution of the fibers within 1 micron pixel size. It's also possible to apply, since we don't stain anything with the polarized light, we can use some staining afterwards. So in principle, you can use fluorescence microscopy. Here, we even don't have to we even don't have to stain. We can process the tissue, washing the tissue in a specific way, and we start to see autofluorescence in the myelinated fiber structures. So, and what you see here is in the background, it's in the mouse brain, it's a polarized light imaging, and here all these rectangular fields are the 3D measurements inside the section based on 2-photon fluorescence microscopy. And so this is a good idea. It works. Another question is, what I haven't addressed yet, we are only talking about some connections, right? It's a structural information. There's a fiber and it runs in that direction. But it doesn't tell you in what direction it communicates. We don't know the type of the connection. So it might be a good idea to find ways to identify the types of neurons. And here's one we are following together with our partners from Amsterdam, from the NIN. And that's a vervet monkey brain sagittal section. And this is the same section afterwards immunohistostained for calcium binding proteins. So you see 3 different colors, and each color here, or each colored neuron, represents a different kind of calcium binding protein, parvalbumin, calbindin, and calretinine. So it is possible to do this staining, and what do you gain? You can see here, right? Driving towards the LGN. On the left-hand side, you see the fiber structures, and here you see the distribution of selected, very specific types of neurons. Right? And this is the way to go, one way to go ahead, trying to characterize and typicize the different connections. Of course, we have applied this also for the human brain very recently, and it also works. Immunohistochemistry is always tricky because the tissue has to be as fresh as possible, which means no long formalin fixation times. And usually for the human brain, we don't have this available. So whenever you want to do something like this, you really have to take care of having fresh brain, only a few weeks of fixation available, then the staining works. And then lastly, from our own ideas here, we generated a tour d'Europe based on measurements. So we cut a mouse brain. here and send it to Sweden, Austria, Spain, different regions in Germany to combine the polarized light stuff with X-ray, to combine it with different types of electron microscopy. The same tissue, it's always the same tissue, right? And eventually it works, right? So you can do it. So we did it in a way like having the polarized light data, send it to Gothenburg. They prepared the brain for EM, but kept it in this kind of resin way, send it to Vienna. They did X-ray imaging to see whether the staining was good. And according to the X-ray, we defined the cutting angles and the cutting region and sent the cuts, the individual cuts to Zeiss with a multi-SEM beam, so having many measurements at the same time with an electron microscope, or sending to Madrid, having a FIB-SEM available just to see whether it is possible or not. And finally, it comes with something like this. This is a poorly aligned, but it's an aligned data set starting with the polarized light. Here on the left-hand side, it's the X-ray. And here, this is the region where we applied the different techniques of electron microscopy. And it is possible. It's not possible to do it for the entire brain, but it's a starting point. So what did we achieve so far? To sum up a little bit, I've given you a lot of information up to here. So I see currently two branches of connectome research, right? The first one is the large cohort studies based on whole brain in vivo diffusion MRI. This is set, this is working, there are infrastructures available. Of course, you're doing microscopic resolution, you're doing white matter imaging, large distance imaging is possible. Tractography has some limitations still, but we can target inter-subject variability easily, having a lot of brains available from the individuals. And then there's a second branch based on small numbers of brains, small samples usually, post-mortem studies, different contrast mechanisms. Here we reach the micrometer to nanometer resolution. They are mostly working on gray matter, but polarized light imaging, for example, I've shown you is able to cover more both, so gray and white matter. Tractography is challenging for other regions, imperfect alignment of the sections, and additional analysis steps like structured tensor analysis. And finally, of course, we have to deal with big data analytics, high performance computing, right? But there are worldwide, not only in Jülich, of course, I'm biased, I've shown you our work, but there's worldwide a lot of work ongoing to make multimodal correlative imaging of the same tissue sample possible. So now coming to my personal wish list for the next couple of years. First of all, we have to push further this correlative imaging idea, right? And in this context, I would like to assess the directionality of connections. So try to find the different types involved in specific networks. We need to correlate the axonal network architecture with the neural cellular architecture. So of course, these are different views of the same thing, but how can we really bring this together and even go to dendritic architecture? So in this case, we really have to go for higher resolution fluorescence and even electron microscopy. We have to think about tractography algorithms. There are new approaches available also based on machine learning, and it's getting better and better. The more information we have, the more we include anatomy to the algorithms, the better we will come up with a diffusion MRI signal reconstruction. So a second point I would like to highlight is the atlasing idea. So whenever we want to share all this data and want to analyze the data, if you want to make an analysis, you need to be sure that the data you're using is curated, is seen by an expert, and is provided in an understandable and easy way. So we need Atlas frameworks. There are some on the market, and they are being developed, but currently you see here some, there some, and they all are somehow different from each other. So what we have to reach in the end is some interoperability of these atlases and information for all the people who want to make analysis and want to make use of this, this data set. So third, I just mentioned this, so we have to generate and share more data. We really have to share data. Neuroscience is not known as a community that easily share data. And I think once we provide infrastructures for this, the situation might improve, and that's our hope. And finally, I haven't addressed this in any case. What about functional imaging, right? So dynamics of the brain, plasticity, this needs to be integrated as well. And of course, I would like to see some kind of these correlations between real functional imaging and structural imaging. So I'm just 30 seconds over time. I would like to mention that there is a European kind of approach to an atlas, an interactive atlas. It is called eBrains. You can find it under ebrains.eu. And there you can find data. You can also share your data. You can find tools, how to share data, how to align your data to this. And this is an interactive kind of developing atlas. It is interactive in terms of, I skipped this, in terms of having Python interfaces available, so you can use this interface for your own analysis based on Python. It's easy to use. There's a nice tutorial. Please have a look and maybe try it out, and if it works for you, and it would be beneficial for all of us. And with this, I would like to thank a lot of people who provided data and videos, images, and to this talk. And I would like to close with this cartoon video. And I'm really looking forward to inspecting the brain with all of you. Looking for all these surprises we are expecting. And eventually realizing that you're not the first. Thank you very much.
That fascinating talk. Questions from the audience? Yes.
Thanks a lot for your talk. That was really, really fascinating and some incredible pictures there. My question is more philosophical, perhaps. I'm wondering, as for you as a researcher, as a scientist, now that we have all this technology and that we can really zoom in on the brain so much, what do you think that will eventually tell us about behavior in the human mind?
Oh. Yeah. So I think, yes, if we are able to correlate directly functional imaging with structural imaging. So what's currently really missing is that you have an object, a subject in vivo, which donates its brain, her brain after death, so that you can have all these different steps from functional in vivo, structural in vivo, to down to the microscope data. Then I think some things might become more elusive, but I don't know. This is future work. We try to get these cohorts available, so there are a lot of elderly people available. we also see we have I have some collaboration with colleagues from Tours in France, and they are monitoring and supervising about 50 elderly people. They are willing to donate their brain afterwards, and they are being scanned all day on a regular basis at Neurospin. And we see they are not dying with the statistics because they are well monitored as well. That's a good thing, right? So we are all patient, but I think this is really what we have to do. If you want to understand some function, you need to have this direct correlation between in the same brain eventually. But I'm pretty sure also the variability from one brain to the other cannot be addressed in an adequate way just by a small number of people, especially limited by the postmortem scans. So doing one, I didn't mention this, doing one polarized light imaging brain means for us an effort of 3 years currently. That's a long time.
Yeah, I think his talk is fascinating. My question is based on basically for all the postmortem studies, you get a lot of findings. Right? How much of that findings can be translated to the downstream vivo diffusion MRI analysis?
Yeah, that's a good question. I think using machine learning models, so thinking about some kind of foundational brain model based on Atlas data, I think we can translate a lot of things into the clinics as well. And we, independent of this, another piece that is already happening, actually happening at hospitals in Marseille, for example, the physicists, they take out our data and calculate some simulations for epileptic patients, being able to predict based on the individual data, based on the anatomy, where's the lesion focus of the epilepsy of each individual. And that might be, I think, the next really good step, using this kind of information and try to do a personalized kind of medicine.
Yeah, I think specifically I'm interested in two direct applications. One is like you can do the digital twins for
Yes.
brain. The second thing is I'm interested basically how much information I can use to directly build the brand atlas. And basically like the vivo, you have a vivo diffusion MRI, right? The current analysis is based on the vivo MRI, the diffusion MRI to construct it. But that if you have the small quantum informations, I can easily build a much better kinds of atlas for the downstream analysis. Because you can, in certain degree, you can directly dramatically reduce the false positive for the fiber tracking for the downstream diffusion weighted images, right? That's my, when I saw all these findings, this is my directed two major applications for that. What's your opinion?
Yes, so the direct applications, I mean, if you provide access to the labeled and curated anatomical information in the process of brain surgery, for example, right? It is useful to have a general kind of high microscopic view during the surgery available on the reality, right? So this kind of fusion augmented reality applications, I think this is where we are not too far away from. that might be one of the applications of
One
this.
very quick question, which I will selfishly ask. Earlier in the talk, you mentioned about two regions or more that share similar structure. I'd like to ask, what are the features you're looking for when you talk about similarity? What is a metric that usually you use?
Yeah, so first of all, we look for the distribution of cell bodies. So it's Nissl based on Nissl staining. But using this contrastive learning showed us that applied to the fiber measurements, showed that we can also do some kind of mapping and regional clustering based on the information of the fibers as well. They do not necessarily have to agree, but we don't know yet.
Thank you very much. We have to move on to the next session, and thank you very much again, Marcus.
Thank you very much.